concentration cells with the transference and without transference. So first let us see what are concentration cells. Electrochemical cells where the same electrode and electrolyte are used but with different concentrations in the two half cells. Electrochemical cells. What is electrochemical cells? First, cell is a device which converts chemical energy into electrical energy. That's called cell. Electrochemical cell, conversion of electrical energy into conversion of chemical energy into electrochemical cell. So such kind of cell where the same electrode and electrolyte are used, same electrodes, for example copper electrode, both electrodes are copper. Electrolyte, for example copper sulfate, copper rod is dipped in copper sulfate, iron rod is dipped in iron sulfate or iron chloride, zinc rod is dipped in zinc sulfate solution. So like that, same kind of electrodes, but same kind of electrodes and same electrolytes, but the concentration of electrolytes are different in the two half cells. So such kind of cells are called electrochemical cells or the concentration cells, electrochemical concentration cells. The potential difference arises due to the concentration gradient. The potential difference comes because of the difference in the concentrations of the same solutions. Suppose if the concentrations are not same, then there is no point of potential difference at all. So that's about the concentration cells. So come EMF of a concentration cell. EMF means electromotive force. In other words, the force, the unseen force which motivates the electrons to move, it's called electromotive force. So the electromotive force is equal to nRT, RT by NF into log of C1 by C2. R is Rydberg constant, T is temperature, N is number of moles or number of electrons, F is Faraday, and C1 is concentration of the solution in one half cell, and C2 is the concentration of the solution, same solution in another half cell. So E cell is equal to NFRT. Similarly, E cell is equal to NF by RT log of C1 by C2. So C1, C2 concentrations in the two half cells. R is gas constant, T is temperature, F is Faraday's constant. This is EMF measurement formula. EMF E cell is equal to RT by NF log of C1 by C2, or E cell is equal to RT by NF log of C2 by C1. So types of concentration cells. There are two types of concentration cells, with the transference and without transference. With the transference means with the transfer of the matter from one half cell compartment to another half cell compartment. Without transference means there is no transfer of the ions. So definition with the transference. It involves the migration of ions. Ions are migrated across the membrane or salt bridge, contributing to the cell's EMF. It involves the migration of ions across the membrane or solid bridge contributing to the cell's EMF. So in case of cells, concentration cells without transference, there is no migration of ions across the membrane or salt bridge contributing to the cell's EMF. So the working principle includes the potential is generated by both ionic concentration differences and ion migration. The working principle includes the potential is generated by both the ionic concentration differences. Just because of the concentration differences, potential is generated. So this potential is the key point to this entire topic. And anions move towards the higher concentration side and cations towards the lower concentration side. EMF calculation is equal to E cell is equal to E electrolyte plus E transference. So E cell is equal to E electrolyte plus E transference. E represents EMF or electromotive force. Concentration cells with the transference. So no significant ion migration contributes without transference. Concentration cells without transference, in this case, no significant ion migration contributes to the EMF. So the potential arises only due to the difference in concentration between two half cells. Difference in concentrations between two half cells. Working principle: a sensitive barrier or membrane is used to restrict ion migration. EMF depends only on concentration differences. EMF depends upon the concentration differences. EMF is equal to E cell, that is equal to RT by NF log of C1 by C2, or RT by NF log of C2 by C1, C2 by C1, or NF by RT log of C1, C2 by C1. So this is the formula to calculate the EMF. So the differences between with and without transference cells. The aspect is ion movement. The ion movement in case of with the transference involves ion migration, but without transference, no ion migration across cells. EMF contribution: concentration plus transference, and EMF contribution without transference, concentration only. So selectivity: in case of with the transference, no selective barrier. In case of without transference, requires a selective barrier. Example: with the transference, solid bridge cells. Without transference, cells with ion exchange. Cells with ion exchange. So let us see some examples of concentration cells with the transference. Copper electrode with different concentrations of copper sulfate in each half cell, and a solid bridge allow ion migration, so contributing to the EMF. So the reaction is copper 2 plus takes 2 electrons and gives copper solid, and copper solid generating 2 electrons and gives copper 2 plus ions. So this process is in equilibrium. In equilibrium, copper taking the 2 electrons, copper which is in the liquid state, that is copper 2 plus ions, and taking the 2 electrons from the external source and converts into copper solid. So this is called reduction. Copper solid splits into copper 2 plus and 2 electrons. So this is called oxidation. This is called oxidation. Now, so examples of concentration cells without transference. Hydrogen gas electrode with different concentrations of HCl separated by a membrane. So no ion migration contributes to the EMF. So here hydrogen electrode represents H plus ions taking the electrons and converting to hydrogen gas. Similarly, H plus ions, hydrogen gas splits into hydrogen ions and electrons. These two processes are in equilibrium, and this is about without transference. So, so myself, Dr. M. David Raju, Associate Professor, Department of Chemistry, PP Siddhartha College of Arts and Science. So thank you very much.